Occasionally, you will be asked to collect additional samples at a specific sampling site to be used for quality control purposes. These samples are used to verify the accuracy and reliability of the data collected throughout the season. At the site selected for quality control, you will have an extra bottle each for collecting chlorophyll, nutrient, and microcystin samples. You do not need to do a quality control sample for algae. One container for each sample type will be labeled with just the site number, like usual, and the second container will be labeled with the site number and the word split. To prepare a split sample, fill the collecting jug as usual. Then pour it into the 2 sample collection bottles for chlorophyll by going back and forth between the bottles, literally splitting the sample as you pour until both jars are filled to the shoulders. Use the remaining water in the collecting jug and repeat this procedure to create split samples for the small nutrient jars and the microcystin vials. After you have returned to Clamp headquarters, find the sample collection jars, 1 each, for chlorophyll and nutrients labeled blank sample. Find the jug of deionized water and rinse the collection jug you used out on the lake with a small amount of deionized water 3 times. Then fill the collecting jug with the remaining deionized water. Pour the deionized water from the collecting jug into each of the bottles labeled blank. Place the nutrient and algae samples, including splits, into the refrigerator. Place the microcystin vials, including the split, into the freezer. Filter the split chlorophyll sample as you would the other samples. Use the Secchi disc reading to determine the amount to filter, and be sure to write this on the plastic chlorophyll vial just like the other samples. Filter 200 ml from the blank chlorophyll sample following the same procedure you use for the regular samples. Label the plastic chlorophyll vial with 200 ml and mark the sample location as blank. Place all filtered chlorophyll samples, including split and blank, in plastic vials and store in the freezer as you normally do. Be sure to put remaining chlorophyll and blank water samples in the refrigerator. Do not dump them out.